So a project that we started in our lab was to evaluate if machine learning techniques could help us predict Legionella pneumophila serogroups from short read sequences. So just for some background information, the genus Legionella, they're gram-negative bacteria commonly found in freshwater and human-made water systems. To date, there are more than 60 species in this genus, and potentially all of them can cause Legionnaire's disease or a less severe form, which would be called Pontiatic fever. And in 1976, the first documented outbreak of Legionella was associated to Legionella pneumophila, and the genus Legionella was paying homage to that first outbreak from the Legionnaires' conference where it occurred. The risk group for this infection is usually males over the age of 65 with some type of underlying respiratory condition, such as those commonly found in smokers. Symptoms are usually ranging from fever, muscle aches, cough, and the onset is usually 2 to 10 days after exposure. LD is usually the fatal form of this, leading to a severe pneumonia. There are various plans in place to help people identify Legionella since it is an environmental pathogen. One of these available is ASHRAE, which helps individuals how to detect Legionella in their water supply and remediate this issue. Legionella and infections usually is caused by this mode of transmission. Legionella will be present in your water system. There's amplification and then aerosolization with the bacteria, usually inhaled into an individual who's susceptible to this infection. That's usually from decorative water fountains, whirlpools, spas, cooling towers. So we see increase in these outbreaks during the summer months of the season. Currently, Legionella has been the leading cause of waterborne outbreaks in the US with an 200% increase over the last decade. We typically respond to about 8 outbreaks via an EpiAid over the year. And since this pathogen is environmental, it's important to identify the source, which we call source attribution, to the clinical. And this is usually done through a series of various methods, such as sequence-based typing, which is equivalent to multi-locus sequence typing in other pathogens with 7 housekeeping genes, monoclonal antibodies, and pulse-filled gel electrophoresis, and serogroup, which we will focus on for the remainder of this talk. So before the era of whole genome sequencing, this was our traditional workflow to identify the serogroup for Legionella pneumophila isolate. If it was a water sample, we would filter it, then wait for about 7 days for growth, and then using slide agglutination or direct fluorescence antibody assay, determine its serogroup. And the same would be, as you can see to the right of your screen, similar process for a lung tissue or sputum specimen. So currently you're looking about 7 to 9 days before you can actually identify the serogroup, which actually is a long length of time for that individual who's sick to identify these additional characteristics features. But now that we're all in the era of whole genome sequencing, we're hoping to use this technique to reduce the number of traditional molecular detection and characterization assays in order to use a single experiment, WGS, to extract this information from the sequencing data. So just for a little information on serotyping, for Legionella pneumophila, this is usually the most common species associated with LD infections, representing about 90% of the cases. And of those, most are contributed to Legionella pneumophila serogroup 1. I would like to, at this time, just give you a little workflow of what we would do in the lab with our assays in order to determine serogrouping. So you can kind of get well, for us, it was to get an appreciation of doing this from whole genome sequencing, since this is still a new technique applied in our lab. So initially, for site agglutination, we have a series of antiserum pools, 1, 2, 3, 4, and 11, and they are actually mix of a combination of different serogroups. And then after the SAT pool is ran, the individual SAT is run for that serogroup for a confirmatory result. Following up with direct fluorescence antibody, which is more specific in serogroup determination than slide agglutinase detection assay. We run all 3 of these. So just for a little simple workflow, we would run initial the pool, and we'll say that, excuse me, I'm sorry about that. We would run the pool, and for this example, we say that pool 2 was indication with serogroup 6 running that individually, and we have a scale from 1 to 4 plus on how we determine the cloudiness of the reactivity of the antigens on our slide. And as you can see from the DFA example, we would say that this isolate X is serogroup 6. However, this is what our results look like. Not oftentimes, but sometimes, where we see cross-reactivity within the pools, as well as when we run our individual serogroups as well. You see cross-reactivity from various serogroups. And even with the DFA, we were still unable to determine this isolate serogroup, so we determined it to be an L4/10. Now, this is just one example of just showing some of the issues with the cross-reactivity. There's also a number of reagents that we have to keep with QC, making sure they're maintained to identify the serogroups respectively. So imagine that we already have the 5 antiserum pool vials that we have in place, and then each of those have their individual serogroup vials that you have to keep. So you're just keeping a track of a number of reagents in place. I do apologize if you keep hearing a little notification. That is my email going off. I apologize.
Shatavia, you're doing great. I'm sorry that we caught you off guard. For everyone who doesn't know, we did not make an actual calendar event with Shatavia, and we caught her off guard by having by not giving her Eastern time zone. And you're a real trooper for doing this, and we really appreciate it.
Yeah, no worry. I'm just hoping that the sound of the email isn't really hurting anyone's ears. If you have ear pods in, I know how sometimes that can be annoying. So I do apologize.
It's all good. We're kind of used to this, including kids running in the rooms.
Okay. Well, thank you. I appreciate that. Now okay. So yes, before I showed the cross-reactivity that was going on with various serogroups for that particular isolate. So I hope at this time I've been able to convey that the classical method that we were using was time-consuming and complicated. You'd have a workflow of you going from SAT pools to the specific serogroups to the direct serogroups and the potential for cross-reactivity. It's just an assay that if we could replace it with whole genome sequencing, definitely would like to. So in 2000, the LPS region, which has been associated with the serogroup, was sequenced. The LPS region has an core antigen, an O antigen, and a core component that has variability, which has actually been very helpful in serogroup determination. 8 years later, there was a more in-depth study with this region that identified not only the same genes from the initial study, but that they were able to identify 2 genes, WZM and WZT, that were specific to LP1 isolates, which could be used for a diagnostic feature to determine LP1 versus LP2 through 17. These 2 genes are also used in current real-time PCR assays to determine this as well. So at this point in time, you would be able to at least decipher your isolate into 2 groups, LP1 versus LP2 through 17. However, 5 years ago, there was a study that was released that actually showed there are variants of this WZM and WZ2 and other serogroups. So initially assuming that the WZT and WZM were specific to just LP1 was not necessarily accurate. However, the variant alleles of those genes in LP1 are specific, which still makes the real-time PCR assays that individuals use still applicable to determining the serogroup for just those isolates that are LP1. So with this information and from the other studies where they had multiple serogroups, but they didn't necessarily encompass all of them, we took the approach and some sequences from some isolates from our collection to sequence 183 Legionella pneumophila isolates representing all serogroups. And utilizing machine learning techniques to capture this information from short read sequences. Machine learning was definitely an approach in our lab that we hadn't had any experience with at the time and didn't have any experience with. So we came across this application called H2O, which was very helpful for us for learning how to code and build into machine learning applications and determine if we were using the appropriate methods for our data set. So our initial question was, does the LPS biosynthesis region have a combination of SNPs that would be beneficial for machine learning? Again, at that time, about a year or two ago, we were just wondering, why hasn't anyone done this before? Maybe they knew that the sequencing data just showed that it was not possible to separate the isolates from the LPS by themselves. Maybe no one had all the serogroups represented in their collection. We weren't actually sure why we didn't see anything in the literature about this, but we thought it'd be a great thing to pursue. We used a combination of bioinformatics techniques and the machine learning component from H2O, the AutoML application, for this work. So here's a workflow of our methodology was to map all the reads to the LP1 reference genome, and within the genome alignments, extract out the 32 genes that were identified as being associated with the LPS region, as well as the SNPs within those locations. If a site was below 20x coverage, we masked it with an N, and a gene also was considered missing if it was less than 80% coverage. And then evaluating the H2O AutoML application, we use 50 isolates in this data set just to initially test the application and understand how to use the algorithm appropriately. And we saw that there was promising results. So we actually went back and took all the isolates and applied it to an R version that we were able to build with random forest and carriage package using a k-fold validation of 10. Then we would produce our predictions and compare those to the DFA results to determine the model's accuracy. So from the H2O application with the 50 isolates, we saw that the random forest method was the most appropriate. The nice thing about this application is that it will run all these classification models for you without having to code for each one of them, which we thought was beneficial at the time. So even with the 40% error rate, we thought that was okay, but then when we looked at the cross-validation for each of the sets, 3 of the 5 actually were above 70%, which is typically was above the threshold of 70%, which is typically a considerable threshold for real world data. So we thought we can continue on with our analysis with this approach. So now applying it to a larger set of various isolates, we asked our question again, and we said, well, yes, kind of, you see a signal within the LPS region using the SNPs. So this could be an indicative region for prediction for LPS serogroups from short read sequences. There are some serogroups that did not perform well, and later on in the talk, we will highlight why that potentially is. Also, I'd like to note, we also included Laplace smoothing, or also known as additive smoothing, which is just including a single occurrence of each nucleotide type, A, T, C, G, T, or N, at that site in the matrix. So for those of you, I'm not sure who are all familiar with machine learning and the random forest approach, I tried to put together a small example of how this would be represented, which random forest constitutes of a magnitude of decision trees. So just for kind of a scaled down version, if you had a question, are you a superhero or a villain? So for our superhero, we have Spider-Man, and our villain, a very unfriendly character. And our features are cape, mask, suit color, and sidekick. And for our data set, for our sequences, our sites would have been our features. So at each site, you would determine the types of decisions you have for those features you see for the superhero or villain. It's yes or no, not red or red. And then going back to the sequencing data set, it would have been of one of those nucleotide types. And then you associate your classifier, which here we're saying superhero is associated with these characteristic features and the villain is associated with the others. You would train your model not just on one image, but thousands of images, and then you could have a test set. So now we have a test set of 2 separate images, and we supply those to the model. And you ask, and the model's saying, is it a superhero or a villain as a classifier? Hopefully, with successful training and your robust data set of training method in your training method, your classifier would show correctly. So at this point, we would hope that we would identify the superhero correctly and the villain correctly. This was a very high level approach to machine learning that I tried to explain. So I probably have left out quite a bit of the mathematics behind, and I do apologize if I just wanted to give you at least a little background about that. So now jumping back to the data set, looking at the R version of our model, we used 180 isolates. We removed 3 isolates because there was only a single occurrence of that isolate in the data set, which were 11, 16, and 17. So as you can see, highlighted in the gold, several isolates have an accuracy above 70%. There are a few that are below that, and again, we will show hopefully why that has occurred, but we think that we've done pretty well with our prediction model, that we've at least been able to increase the classifying group past the real-time PCR assays that are out there from LP1, from an isolate being either determined as LP1 or LP2 through 17, that at least we're able to identify 7 different categories of an LP serogroup. So we wanted to test our model on blind data, and we had 3 separate data sets. The blue is representative of a correct prediction, the red is incorrect, and the size of the circle is indicative of the number of isolates that we had for that particular serogroup. So in this data set, we had approximately 370 isolates that were LP1. The accuracy for this data set was 95%. You can see that some of the serogroups had 100% accuracy, while there were others that were more problematic, such as 2, 8, 10, 12, and 15. So we wanted to test our model's robustness, that since we trained it with data that we sequenced, and then we now have tested it with data that we sequenced, we wanted to see how the model would perform with sequencing data that was not generated in-house. As we all have known, or anyone has experienced with sequencing data, there's always a little inherent variability, even just when you have the same library prep ran on the same instrument. So to test this out, our model still holds up with different data sets generated externally. Our prediction accuracy was at 80%, 2, 3, and 1 had high prediction accuracies. Again, the same programmatic serogroups show issues, 5, 8, 10. And our final external data set was at 85% accuracy. Again, those same problematic serogroups started to render themselves again. And we also observed that when it mispredicted, it wasn't random. It usually was predicting the same serogroups. So if it was supposed to be an LP5, it would be predicted as an 8 or a 10. So we weren't really sure about what was going on with this. So we did take a deep dive into the data from a pairwise percent identity analysis. And we asked, are the LPS biosynthesis sequences of each serogroup more similar to themselves and other serogroups? Since we saw this predictions from LP5 to LP10, there was no random nature to it. And as you can see, some of the serogroups have a strong correlation to themselves. The expectation would have been that you would see a diagonal on this heat map that the isolates, the serogroups would be correlated within themselves. However, you see that that doesn't necessarily fit this case, especially for LP5. It doesn't even look like itself from this approach. Also, another thing to highlight is serogroup 6 and 12. Every time we would use for the serogroup 6, it always would be predicted as a 12. And then for 12s, they would be predicted as 6s. And we looked at the percent identities. These were at a 99% identity and percent similarity. So taking another layer to look at the isolates, well, maybe there was some misclassification from the DFA. We have shown that there was a cross-reactivity that could have occurred, or maybe someone may have mistakenly just wrote down the serogroups indicator for our classifier. So we just looked at the isolates that we did classify for each serogroup and do an intra-comparison. As you can see from serogroup 1 and 3, there's a strong signal that all the isolates that are associated with that classifier correlate with each other. But if you look at serogroup 5, you see that it's a wide range of genetic diversity within that serogroup. I'd like to point out within that, yes, my mouse works, 2, 3, 6, 8, they all seem to share a similar pattern. So we started to think maybe there's potential subgroups within certain serogroups. And then going on, looking at those other problematic serogroups, you see similar patterns again, that maybe there's some potential subgroups within these serogroups. But then we go back to the SAT information, you see that there were type strains for each of the serogroups that did not necessarily react the same way all the time. So for instance, if you had an LP4LA, that would not react with an LP4 port assay. So maybe from the sequencing data, we're also able to get to this level of granularity with the SAT type strain information. So perhaps we may just need to go back and reform our classifier to include this level of information as well. So to keep in the spirit with bioinformatics and working together and transparency and interoperability, we put all the code that was generated for this project on our CDC GitHub page, as well as we created a Docker container for this work that we'd love to have feedback on to improve upon the model and see how we can increase the prediction accuracy for some of those poor performing serogroups, as well as some that show large genetic diversity. So just some takeaway points from this talk was that I hope that I was able to convey that the traditional methods for serological based typing were time consuming and varied in reproducibility, not only due to subjectivity of the data interpretation, but also from the QC of reagents and other compounding factors. And we were able to identify genetic variation within some serogroups, which actually may have impacted the performance of the model, such as those in LP5, but also noting that some of these serogroups were only represented by 3 or 4 isolates. So maybe we did not have enough in our training set, so incorporating different additional sequences may help in our prediction accuracy. Positively for this work, we were able to accurately predict several serogroups, 1 through 4, 7, 9, and 13. So we were able to improve upon identifying an isolate serogroup from the traditional LP1, non-LP1 to at least LP1, 2, 3, or 4, 7, or 9. And finally, that this work was available for in a Docker container or bioinformatics analysis. At this moment, I definitely would like to point out the members of the lab and the lead microbiologist who was on this work, Natalia Kozak, as well as John Fan from our scientific computing group here on campus who helped with the Docker container, which took our entire pipeline and put it inside a single component, and our 2 external collaborators from the Arizona Department of State of Public Health and the New York State Department of Public Health. Also, I would like to note that the findings and conclusions in this work are those of the author and do not necessarily represent the official position of the Centers for Disease Control and Prevention. I have listed the references for the images that were used in this work as well. Thank you.
Thank you. That was really great. And I'm gonna click my applause icon because I thought it was really great too.
Awesome.
And, And again, thank you so much. I know that the time change was difficult, and thank you so much for jumping on the talk.
No, I appreciate. Oh, were there any questions? I do have a couple.
Yes, I have a few questions I'm going to read to you.
Okay.
So, and we've aggregated them, so you don't have to worry about the chat.
And I was gonna say, I do apologize for not putting my contact information, but I'm on the Slack if you want to send anything directly to me.
Awesome. So a few questions. The first question, hopefully this is an easy one, was what is H2O.ai? I don't think that, at least I haven't heard about it before.
H2O.ai is a machine learning package that was available, and just from talking with the various collaborators and colleagues that they have used it in their work, I hadn't heard of it either until someone told me about it. So it still seems like it's one of those well-kept secrets, but I definitely would recommend if you have the time. I know that it's available in Python and R. I'm not sure if it's available in other languages, but check it out if you have time.
Awesome. Okay, next question is, I apologize for reading off my phone off to the side.
Okay.
So this still requires growing the bacteria and then sequencing it, which could take days. Have you thought about or looked into whether this could be applied to, your technique could be applied to metagenomic sequencing, removing the need to grow this strain initially?
So we have tried to do metagenomics separately to the machine learning, but from just initial sequencing runs, Legionella is very little in the metagenomics data set. And we actually see, and that's just sequencing from an environmental side. We haven't really, and most of the times when we do sequence metagenomics, 60 to 70% of those samples we aren't able to classify. So I feel like we have a long way to go with understanding our metagenomic samples before we can apply it to the machine learning aspect, for sure. But definitely, I believe it's something we would like to get into.
Very cool. And one last question. Have you considered taking the entire genome as input, such as k-mers, for your classification model, as of now you specifically focus on a subset?
No, initially we tried the higher BAPS method to see if the sequences would recover the serogroup information, but what we saw was a phylogenetic signal was more clustering the isolates. So if they were from a particular outbreak or just from a particular year and time, those would cluster versus the serogroup themselves. So if there's a way to, I guess, remove that phylogenetic signal to reclassify for serogroups, that would be helpful. But definitely that's something to try with the k-mers. I haven't thought of that before, but this is never an approach to tackle. I think you're muted.
You're right. Thank you. One last question from Zoom. How was the random forest variability accounted for in the model? And I guess the follow-up question is the nodes of the tree classifier are SNPs. So could you comment on that?
Yes, the sites were SNPs, and to account for that, which I hope we did correctly, so as I say, we're still learning how to apply machine learning appropriately, was just to add a single occurrence for each nucleotide at each site or feature in the model. That may not have been appropriate, but that was our initial test, was to at least make sure that all sites saw every possible state or decision for that category. I don't know if that necessarily addressed the question directly, but that's.
I think so, in my opinion. I don't know what your schedule is like right now. You've basically told me offline that you've been pretty hectic over there with COVID, but if you do have time, we're doing follow-up questions or just conversations on Discord, or else, as Shatavia noted, you can catch her on Slack later on too. So thank you very much.
Yeah, thank you. Yeah, I actually have a meeting in like 25 minutes, so I'm glad. I was like, oh, it's at 4 p.m. I got time. But I do appreciate everybody hanging in there with me, and I'm getting through the talking, kind of fumbling, but had half COVID on the brain. I hadn't fully switched into the Legionella mindset just yet, but hopefully I did my best. But I definitely would love feedback on the model and how to improve it. Like I said before, machine learning was something we had never done before. So if there's some machine learning experts who could help, definitely would love to hear from you guys. And thank you all for your time. And during this time, just take care of yourselves and your friends and family and be safe.
Thanks so much, Shatavia.
Thanks.